All right, well, let's get started. I want to welcome everyone to the OBSSR Directors webinar for October. It's entitled, Is Brain Concordance Linked with Therapeutic Alliance in Pain Relief? Hyperscan fMRI applied to decipher the brain circuitry of patient-clinician interactions. I'm Bill Riley, the Director of the Office of Behavioral and Social Sciences Research here at the NIH. And before I introduce today's speaker, just a few housekeeping items. Today's webinar is being recorded, and that recording will be available on the OBSSR website in approximately a month from now. Today's presentation will also be followed by a question and answer session. All the phone lines are muted during the webinar, so if you have questions or comments, they won't be taken by phone, but instead through the Q&A feature that you should be able to see on the right side of your screen. So all these are set up slightly differently on different computers, but you should see a Q&A or a chat space where you can actually put in information and ask a question that way. And if you click on the Q&A to the screen and select all panelists, you can type in your question and hit send. And then my colleague Bill Elwood will facilitate the Q&A session when we get to that point. So today's speaker, Vitaly Napadow, is Associate Professor at the Martino Center for Biomedical Imaging at Massachusetts General Hospital and the Harvard Medical School, and the Director of the Center for Integrated Pain Neuroimaging. Over the past 15 years, he's applied his biomedical engineering background in neuroscience and neuroimaging research aimed at improving treatment options for patients suffering from chronic pain. Dr. Napadow's laboratory has pioneered the application of non-invasive neuroimaging techniques to better understand the brain circuitry underlying aversive perceptual states such as pain, nausea, and itch, and to better understand how non-pharmacological therapies such as CBT and acupuncture ameliorate these states. Dr. Napadow is the co-president of the Society of Acupuncture Research, more than 120 publications in leading peer-reviewed journals in the fields of pain and in neuroscience. So Vitaly, thank you so much for taking the time to present at the OBSSR's Directors webinar series, and I'll turn it over to you.
Absolutely. Thank you very much. I hope everyone can hear me. I have a quick question before I get started. Can you see my mouse when I move on the screen or not? I just need to know that as I go on.
We can, I think, Vitaly. Do it again for us just so we see. Yes. Yep, we see it. Yep.
Okay. Excellent. All right. Great. Well, it's very exciting to be here, and this is new research from our laboratory, and we're in process. So some of the results you'll be seeing are indeed preliminary, but I think we do have some interesting findings, and so I'm excited to share some of those with you. So a little bit of background. We know that social interactions between a patient and a clinician do contribute significantly to the quality of care in the healthcare system. In fact, satisfaction scores are currently being used to calculate Medicare reimbursement under the ACA. And empathy training for physicians has been gaining interest. There's prominent programs now available that focus on raising the emotional IQ of clinicians through nonverbal information transfer as well as verbal information transfer to try to really improve these outcomes between clinicians and patients in this dyad. So I'll be talking a lot during this talk about the patient-clinician dyad, and so I want to introduce that concept up front. So nonverbal cues, we know, are also very important to concepts such as perceived empathy. This is a recent study that actually used a really interesting kind of a novel approach with over Amazon that recruited over 1,000 participants and just had people rate the warmth and the competence of clinicians that were in different poses. So you can see sort of on the left are these empathic poses that were done by actors, and then there's unempathic poses on the right. And folks rated them, the empathic poses, as having both higher warmth and higher competence. So I think this was really interesting and also suggestive of the fact that patients can sense their clinicians and can sense the body language and the facial expressions of their clinicians, and this can have meaningful clinical outcomes. So for example, social interactions, the patient-clinician relationship, can also significantly shape how we feel and how we conceptualize pain. So a recent trial of spinal manipulation and exercise for chronic low back pain patients found that the Therapeutic Alliance, which was rated by the patients, was an important predictor of pain outcomes, of analgesia, and an important moderator of the effect. So these these concepts of clinician-patient interaction and therapeutic alliance, which many times are actually attributed to quote-unquote placebo effects, actually can have very meaningful outcomes on patient-report measures such as pain. So interaction style can be experimentally controlled also and can influence symptom severity. This was a study by my co-PI on this project, Ted Kapchuk, which took clinicians or acupuncturists trained to deliver acupuncture to irritable bowel syndrome patients under both an augmented or a limited interaction style, and found that those trained to having an augmented style in the way they interact with the patients had better clinical outcomes. So they were doing the exact same therapy, but if they did them under an augmented type of interaction style, the outcomes were better. And so one thing we are really interested in trying to understand is, why this is the case. So there's definitely a wealth of literature out there that these kinds of interactions can have meaningful consequence for clinical outcomes, and we want to try to better understand why. So our goal is to understand the brain circuitry supporting these concepts like therapeutic alliance. So the neural pathways by which therapeutic alliance influences pain is not well understood. So one question is, why is this understanding important? So first of all, we want to define an objective framework for optimization. So for example, if we want to try to explore different interaction styles, if we want to have more cost-effective clinician training by focusing on the most effective components, we need some sort of optimization variable under which these different trials can be assessed. And one of them is to understand an objective measure, such as understanding concepts like brain concordance and the neural circuitry that's underlying therapeutic alliance. We also, of course, want to reduce clinician burnout. So this is, I think, a gaining problem in the field. There's clinicians that are dropping out of medicine, after going through the rigorous training, and we want to limit that. We want to limit the number of clinicians who are dropping out of medicine, and we want to limit the amount of burnout that clinicians are feeling when they are in the medical system. So this is definitely an important problem that I think is only going to gain in importance as we really try to find ways to treat all of our patients in this country. So there's actually been very few imaging studies that have looked at brain imaging of clinicians in a therapeutic context. One of the studies that we are basing a lot of our work on that was done from the lab was by Karen Jensen, published in 2013, which had an acupuncturist that was, not an acupuncturist, a clinician, in this case actually was a physician, who was laying in a MRI scanner. And while they're laying in the scanner, they're watching to them a patient, which was actually a sham patient. It was a research fellow who was then undergoing pain testing, and the clinician could press buttons in order to reduce the amount of pain that the patient was feeling, and the patient would sort of report back by a facial expression as to how much pain she was feeling, with and without the different treatments. And with this type of experimental setup, you can control a lot of the design in the experiment, but still have a more of an ecologically valid context by which a patient and a clinician might be able to interact, while undergoing brain imaging. And in this case, what was found was that when physicians were treating the patient compared to a no treatment condition, there was an increase in brain activity in brain regions such as the ventrolateral prefrontal cortex, that's the vlPFC, and the temporoparietal junction, the TPJ, which are important components of social mirror circuitries. And I'm going to get into that a little bit later in the talk. So clinical social neuroscience actually has paradoxically typically studied single individuals in isolation, such as a clinician that's being scanned while completing some sort of task where they're, maybe interacting with patients, maybe trying to treat a patient. But it's been done mainly in isolation, where you just have been imaging one individual of that dyad, of that patient-clinician dyad. And so one of our contentions is that understanding the brain circuitry supporting social interactions actually requires two-person methods in an ecologically valid context. And so two-person methods, have been around for a little while. Specifically, there's been some, in the clinical context, specifically there's been some work looking at autonomic nervous system concordance. And this was work by Carl Marcy and co-authors back a little bit more than 10 years ago, which had a clinical setting. So this was 20 adults from a mental health clinic, interview with a psychiatrist, and the psychiatrist was trained to deliver an interview with either a neutral type of interaction style with verbal responses and nonverbal responses in a socially and clinically acceptable manner, including mutual eye gaze, head nodding, these types of features. And this was contrast with a distance style. So the same exact clinician would interview a different patient, and this time with a distance style where the interviewer did not acknowledge the emotional and verbal content of the participants' responses, minimized nonverbal responses, engaged in self-distraction techniques, these types of things in order to have more of a distant relationship with the patient. And so what they found was that when the clinician had more of a distant relationship, there was a decrease in the amount of eye gaze between the two individuals. There was also a decrease in the empathy that was rated by the patients for this type of interaction. And importantly, there was a decrease in the physiological concordance. In this case, it was a skin conductance response. And the investigators were looking at the amount of skin conductance concordance between the two individuals over the period that the interview was happening. So there's not just these sort of subjective ratings that can be affected by these different types of interaction styles, but there's also physiological and objective outcomes of concordance that are also affected by the interaction style. So this was really interesting, and I think these studies were interesting because it's one of the first studies that really looked at these two-person methods where physiological outcomes were evaluated simultaneously, concurrently, in an interacting dyad of a clinician and a patient. So the next step is we wanted to look at if there's also physiological concordance in brain activity, and if so, how does this physiological concordance manifest itself in affecting the types of ratings that the patients and the clinicians are giving towards the quality of the interaction style. So a bit of a definition. So hyperscanning really refers to the synchronous neuroimaging of more than one individual. So it could be two or more individuals that are being scanned at the same time. That's the definition of hyperscanning. And hyperscanning typically has been used to look at brain concordance. So this was an interesting study, dating back a few years, which found that there was an increase of concordance in theta band power and a suppression of alpha band power in parietal electrodes when two individuals were undergoing a task that required really strong cooperation versus weak cooperation. In this case, they were working with pilots from the airline Alitalia. This was a Italian group, and they had them go through a simulator where the plane was going up and takeoff and also then landing from Rome to Pisa, and they basically were looking at brain activity during this flight simulation. This wasn't actually happening in an actual plane when people were flying, so don't worry about that. But these types of EEG hyperscanning experiments have been done in other contexts as well. They've been used to look at brain responses and brain concordance during juggling, during music performance, and other contexts as well. So what we're interested in is actually looking at functional MRI or fMRI hyperscanning, which assesses brain-to-brain communication between individuals for both cortical and, in this case, subcortical concordance as well, and also has improved spatial localization compared to EEG. So EEG definitely has some advantages, such as temporal resolution compared to functional MRI, but functional MRI also has some advantages in terms of the depth of penetration and the spatial localization of where these signals and where concordance is coming from. So one question is how do we apply hyperscanning for fMRI? It's a little bit more difficult to envision. So there has been some studies in trying to actually use very large scanners and try to fit two individuals inside of a single fMRI machine. But this was done for a very specific context, and I would argue it's probably a bit too close for comfort for patient-clinician interactions. So probably, in order to do these types of research, what we really need is two-scanner methods for fMRI hyperscanning. And that's really been the most common approach to fMRI hyperscanning in the field. And so this was actually one of the first studies. You can see this research actually dates back quite a few years. Reed Montague was performing linked fMRI scanning, and in this case there was sender-receiver relationships with different a color task involved, and they were actually doing a competitive game, and there was rewards in terms of juice being delivered to one of the individuals. And so this was kind of, I would say, the birth of functional MRI hyperscanning. But these studies do remain difficult to impart, and there's not very many groups, I would say, even in the world, who are doing this type of hyperscan fMRI research. And particularly for the clinical encounter, that's something that has not been done. So in our study, we wanted to have two separate but synchronized functional MRI scanners. And we asked the question, how is patient-clinician brain concordance associated with therapeutic alliance? We also want to know, how does brain concordance impact analgesia, so clinical outcome? And also, what brain regions demonstrate concordance for this patient-clinician dyadic experience and interaction? So our hypothesized target for this is the social mirror circuitry. So a little bit about mirror neurons. Mirror neurons were discovered in the early 1990s by a group in Italy who found that mirror neurons exist in the monkey motor planning region, and they are thought to code action understanding. So this was a very serendipitous finding where the monkey was outfitted with electrodes in different parts of their brain, and during a lunch break, the experimenter took their lunch and started eating it, and he saw that the monkey's neurons in this specific part of the brain start lighting up, not just when the monkey was reaching for an action, which you can see over here in B, but also when they were watching an experimenter or another confederate doing the same thing. So these are neurons that are responding not just to impact behavior in the individual, but also when observing another individual engaging in similar behavior. So motor mirror neurons are now found in other regions, such as inferior parietal, M1, and also have been investigated in humans, mainly with neuroimaging methods, and have been found in inferior frontal gyrus and ventral premotor cortex, which is the homologue of F5 in the monkey. So interestingly, these mirror neurons also are thought to play a role outside of the motor system. For example, in S1, where you might have a somatosensory response, so you see another individual being touched, and you also activate in the brain regions that's responding to the localization and the intensity of tactile stimuli, such as S1. And also this would extend out to empathy and social mirroring systems, and these have been localized to ventrolateral prefrontal cortex, anterior insula, in addition to the TPJ, the temporoparietal junction that I discussed earlier. So our hypothesis is that the degree to which clinicians' mirror neuron systems accurately reflect patients' own brain responses to clinically relevant stimuli plays a critical role in nonverbal cues and patients' perceptions of therapeutic alliance with the clinician, and ultimately therapeutic efficacy. So this is the hypothesis that we're trying to test in our research studies. So in our setup, we have two MRI scanners, both at 3 Tesla. There's a patient in one scanner over here. There's a clinician in another scanner over here. We have an audio-video link between the two. So we have MRI-compatible video cameras. We have MRI-compatible audio microphones so they can speak to each other if needed. And we have MRI synchronized via an external trigger. So we have laptops with in-house software and adapted fMRI pulse sequences that can be triggered with a TTL pulse that's coming from these laptops and are engaging and starting the MRI machine. We also are dynamically tracking the network delay because all of these signals are being carried over the internet, over a local internet. And as probably all of you have experienced with Skype, sometimes there's these delays which make things very difficult to communicate with another individual. So we wanted to make sure that we are not encroaching into the temporal delay territory where you start to see these delays and that's going to affect communication between the individual. And that was indeed the case, that we were able to track them and make sure that the delay was on the orders of tens of milliseconds, not hundreds of milliseconds. So during the study, we also have the patient is outfitted with an evoked pain device. This is a cuff pain. So the cuff pain device has an air system with a hose that's going over to their leg. There's a cuff that's inflated around their leg, and the amount of pressure that's impacted on the cuff is calibrated in order to achieve a certain level of pain perception by the patient. So that's percept matched and controlled during this experiment. The patient can report their pain ratings with a button box where they rate from no pain to extreme pain. And the clinician, on the other hand, they also have a button box, and this button box controls an electroacupuncture device, which is attached to the patients with needles that are placed just above the cuff, and electricity is applied to the needles, and in order to reduce the evoked pain. So we're for the clinicians, we've chosen to work with acupuncturists as they are very comfortable in working with electroacupuncture, and so this makes it more of an ecologically valid setting for a therapeutic context, which is something we're trying to optimize and maximize here with our MRI setting. So the data that's collected synchronously from both MRI scanners includes physiological monitoring, such as cardiac responses, respiration, and skin conductance, from which we can look at autonomic concordance. We're also collecting, of course, bold functional MRI data from both patients and from clinicians during different phases of the experiment. And again, the clinician and the patient are able to observe the other while undergoing this entire experiment. So this is actually some photos from our setup. So on the left, you see this is the cuff pain device. This is the autonomic monitoring is on a separate laptop over here. MRI control is on a standard system from our Siemens scanners. We have two Siemens 3 Tesla scanners. We also have the audio visual and the network monitoring that's happening here on this laptop, so we can very easily observe and control everything that both the clinician and the patient is viewing it at any given time. So we've built software in order to do this, and we're also able to track the network delays, which again are on the order of tens of milliseconds. So compared to typical MRI experiment, one thing to keep in mind is that hyperscanning fMRI needs a larger team to safely support simultaneous MRI scanning in multiple scanner bays. So that's been a painful lesson that we've been learning along the way too. These are very complicated experiments to pull off. So the way we've designed this study is, and this is kind of the flow through the study, is we have two different conditions, an interaction and a no interaction condition. So any given fibromyalgia patient is randomized to first either complete an interaction condition or first complete the no interaction visit before coming back for another visit where they complete the other. So if they go for the interaction visit, there's a behavioral visit with informed consent, cuff calibration, some boosting that's involved for the electroacupuncture device. There's also an intake that is performed between the clinician and the patient before then going towards the actual MRI scan where they can interact during this therapeutic setting, and we can take all of our measurements. They then return for another visit where they have a no interaction condition, and for this no interaction condition, they're basically the behavioral visit only has the, if it's happening first, it has the informed consent, the cuff calibration and all that in the behavioral visit, and then in another visit, you don't have this intake where the clinician and the patient are able to interact with one another. So you go straight for the MRI setting before then coming back for another visit where they complete the intake and then an MRI interaction. And the clinicians are randomized the same way. So any one clinician will see two patients. Any patient will see two acupuncturists or two clinicians. And so they also are randomized in terms of order to counterbalance the conditions of whether they're first going with an intake patient or a no interaction condition. So during the actual experiment, this is our experimental design. There are blocks of rest where the patient and the clinician are just looking at one another. Then there's a cue that appears with a color that's then placed around the image that they're seeing. So this color square can either be green or it can be red. Green is symbolizing that the acupuncturist in this case is going to treat the patient when they receive the pain, whereas the red is symbolizing that the acupuncturist will not be treating the patient when the patient is receiving the pain. And so in the behavioral sessions, both the clinician, both the acupuncturist and the patient are instructed as to what these cues mean, and this is rehearsed before these runs. In the case of the treatment, we actually randomized them to two different conditions, one with real electroacupuncture, one with sham electroacupuncture. And these, if no difference is found later in the experiment, these of course can be collapsed to improve our sample size. So after this pain period, so during this period, the clinician is pressing one button for treating the patient, a different button in order to control for the motor response for not treating the patient. And the patient is in turn experiencing the cuff pain, which is actually the same exact cuff pain no matter what the treatment condition is, to equilibrate that level. Then there's a rest period, and then that's followed by a rating period where the patient is rating how much pain they experienced, the clinician is rating how much pain they think the patient felt, which was really interesting. And also there's ratings of affect and of how they felt about the experience. So our preliminary results, we've gone through about 11 or 12 dyads now, and are double that. We've gone through double that, but about 11 or 12 acupuncturists and patients. So just so you understand kind of how much data is going into these bar plots that I'm going to show. So this is the ratings of the cuff pain, both by the patients and by the clinicians. So the blue is the treatment condition, the gray is the no treatment condition. And so the difference between these two can really be explained by a placebo effect, because the amount of pain that was delivered, the amount of pressure that was delivered to the patient was identical in this case, and we found no differences so far between the real and the sham electroacupuncture condition. So this can readily be interpreted as a placebo effect. And there's some hint over here that the clinicians are overestimating the efficacy of the treatment compared to the patients, and I'll talk about that in a little bit as well. So first of all, what's interesting is that we found a correlation between the patient's rating of their placebo analgesia versus the clinician's rating of the placebo analgesia. So the more the patients are reporting a reduction in pain, the clinicians are somehow able to read that and are also reporting that they felt that the patient had greater reduction in their pain. So that was really interesting. But if we draw a line, a unity slope line through this, this will be the line. This will be the line that if the data points fell on, there would be an equal amount of estimation of placebo analgesia from the patient and the clinician. And what you can see here is that most of the data points are actually falling on this part of the line, which suggests that clinicians are overestimating the patient's pain reduction with treatment, at least until the analgesia becomes moderately strong. So here they're maybe a little bit more active, more accurate. But this, I think, is really interesting, and as we collect more data, it'll be really interesting to see how this pans out. So these are the brain responses that we're seeing so far in both the patients and the clinicians, and in response to the cuff pain or the treatment phase. So this is the brain response in the patients to their own pain. We see nice activation of S2, posterior insula, sort of contralateral over here to the cuff pain, but also we're seeing interesting activation over here in the temporal parietal junction, the TPJ, and also the ventrolateral prefrontal cortex over here, as well as the anterior insula, these sort of social mirror circuitry areas that I talked about earlier. And what we see in the clinicians is that there's actually stronger activations in these same brain areas, such as ventrolateral prefrontal cortex and TPJ. Similar in the clinician. So if we look at the conjunction map, so the conjunction basically shows us what brain areas are commonly activated in both clinicians and the patients during the same period of time. That's the conjunction. And what we see is, in fact, that there was nice conjunction in the TPJ, the temporoparietal junction, and the ventrolateral prefrontal cortex. So that was interesting. What we also found was that when we looked at the amount of voxels that were co-activated during the treated condition versus the no treated condition, we actually saw that there was a greater amount of, and looking at this as potentially a measure of brain concordance, we found that there was a greater number of voxels co-activated under the treatment condition than the no treatment condition in the ventrolateral prefrontal cortex. And what's more so, the amount of concordance, or this difference between the treated and the no treated condition in individuals, was correlated with the patient's placebo analgesia. So there was sort of this impact of this brain concordance on a clinical outcome metric. In this case, the analgesia that's reported by the patients in the treated versus the no treated condition. So suggesting that there is information transfer in both directions, from the clinician to the patient, as well as from the patient to the clinician. So switching a little bit, can we also link this functional MRI response with automated emotion detections? So emotions can be objectively estimated from feature point tracking of facial videos. And so this is kind of a research field which is really exploding, I think, right now, and gaining an interest for AI, artificial intelligence, and machine learning, as different researchers are trying to read faces of individuals and make certain assessments, either in the commercial realm, the political realm, or even the medical realm. And so what we've done is we've used one of these available softwares called Effectiva, which has a open source library, and so we're able to use their SDK for software development. And so the algorithms basically use facial expressions in order to map towards emotions. And these are built on kind of validated faces, the Ekman faces that maybe a lot of people are familiar with here. And it takes into account, interestingly, cultural display rules, lighting, camera angle, et cetera. So it's pretty robust. We've had good luck with it. And so what we're doing is that we're estimating from the videos that are recording what the emotions are. So how exactly does it do this? It basically uses these control points to, in order to find different patterns of expressions, such as brow furrowing, lid tightening, smiling, these kinds of things. And then it maps those onto discrete emotions. So, for example, here's some, examples of facial expressions that are as part of their library, and part of the feature point detection for these different expressions can then be used to map to different emotions, such as disgust, for example. So what we found that was interesting, actually, and why I bring up disgust, is that in our data, since there's no, as you can see here, there's no pain face, there's no pain emotion. The closest that we found with our data was disgust, which is a combination of nose wrinkling and upper lip raising. And so what we found is that we can take our videos, we then segment them in order to create a larger field of view in order to make it work with our software, and then we do feature point detection by labeling discrete places on the face that are then used to map different expressions, which can then be used to connect and map to different emotions. And so for this, we have a validating kind of a training run scan, a separate scan that we did, where in this case, there's no actual treatment by the clinician. They're just watching each other while the patient is undergoing the pain. And in this case, there's two different conditions. There's a low pain and there's a high pain condition. And then the clinician is just observing the patient undergoing pain. And what we found is that there was an increase in this disgust score when the, so this is basically a dynamic plot of the disgust score, for these different phases, such as, the resting period, the red and the green are the cues that they're seeing, and then the blue is the period when they're actually experiencing the pain. And so this is the video of the patient while they're undergoing this entire experiment and how the disgust score as mapped by the software is tracking over time. So this is it's really interesting. And what we found is that there was a statistically significant increase in disgust score, both during the Q period, so the high pain, which is the P40 or 40 out of 100 pain, versus the non-painful squeezing condition, and for the Qs as well as for the actual experience of the pressure itself, there was an increase in disgust score for the high pain condition. Now, interestingly, this might be a little bit of almost like a reward, where there's actually a decrease in disgust score relative to rest when they're kind of relieved in seeing the P0Q. So that was kind of interesting. And so we also found correlations that the clinician's rating of the patient's pain, so we found the correlations for the clinician's rating of patient pain between disgust scores and the patient's pain ratings. So the greater the amount of pain that's rated by the patient, the greater the disgust score. And so this was the case for clinicians, not for patients. So the clinician's rating of the vicarious pain in the other individual was very nicely linked with the disgust score that's seen on the videos, but not for patients. And you can imagine why this might be the case. There's definitely a lot of variability between individuals. Some people, show a painful face when they experience a pain. Some people do not. The clinician is reading that face. So that's why the clinician's rating of the vicarious pain might be correlated with the disgust score, but the patient's would not. So our conclusions, while preliminary, are, first of all, therapeutic alliance can significantly shape clinical outcomes. Understanding brain circuitry supporting social interactions require two-person methods such as hyperscanning. Physiological concordance might be linked with perceived empathy, and that the concordance between activity in clinicians' mirror neuron systems and patients' brain responses to clinically relevant stimuli may in fact support nonverbal cues and patients' perceptions of therapeutic alliance with the clinician. So we have, we're in the middle of the study, and we have some really interesting and leading data, and it'll be really interesting to see at the end of the day how much of this is borne out. There's still a lot to do. One thing we're acutely aware of is this concept of pseudo-concordance. So from the EEG hyperscanning world, the idea that consistent phase differences between two signals does not necessarily imply covariance between them. So if two oscillators, such as two clocks, are in phase, there might in fact be reciprocal connections between them and sort of concordance based on this, or one clock might be driving another clock, or there might be a third external influence, such as the induced relationship, or this might be completely coincidental. So you can imagine if both the patient and the clinician are actually experiencing a similar task design in these fMRI experiments, they might be having similar responses only because the design is temporally consistent between the two individuals. It's not that there's actually shared information between the two. So we want to be really careful in controlling for this. So for example, this is relevant for our fMRI study too. And so we want to also look at and evaluate correlations in the block-to-block variability in response. So even though the blocks are the same, temporally for the clinician and the patient, you would not necessarily expect to see that when the patient's response is high for any one given block, the response would be higher for the clinician, or vice versa. When it's low, it's low. So if we see a correlation between the patient's and the clinician's response, that would be kind of interesting. And what we found is that there was variability in this concordance between individuals, between dyads, and if there was high concordance in the trial-to-trial variability between individuals, this also was correlated with the patient's placebo analgesia. So that's kind of interesting, and it's something that we need to, I think, explore more, as we collect more data. Another approach is to control for task-induced pseudoconcordance by comparing metrics between real and dummy dyads. So in this case, you would take the concordance estimate and the metric from an actually interacting dyad and compare it to a dummy dyad or a fake dyad, where you have the patient from this dyad compared to a clinician from some wholly separate other dyad, because then obviously they're not interacting with each other through the video and audio link. So ultimately, we want to untangle this brain behavioral loop. The behavior and the aINS response of the patient is coupled with the brain response of the clinician, and in turn, the behavior of the clinician might predict the brain response in the patient. And there's some really nice reviews on this by Uri Hassan and his lab over in Princeton and Konvalinka and groups in Europe. And so we want to take this hyperscanning research, connect it to the clinical level, and really ask questions such as, does Therapeutic Alliance mediate the link between brain activity and real-time assessments of autonomic nervous system response, facial expression, etc. So this research would absolutely not be possible without a large team of individuals. The postdoc leading this work is Dan Michael Ellingson, who's with us from Norway and has really been instrumental in a lot of this research. Kylie Eisenberg has been instrumental as the research assistant. Jungchen Lee and Chungjin Zheng have really been important for running these scans, for devising the software that we're using to run these scans. And without them, there's no way this work would be possible. And also my collaborator and co-PI, Ted Kapchuk. And of course, without the generous support of the National Institutes of Health and several grants that are supporting this. And from both within this country and in Korea, this work would definitely not be possible. So thanks a lot for your attention. And I'm definitely happy to answer some questions in our remaining time.
Great. Thank you, Vitaly. Really nice and much appreciated that you're sharing sort of emerging work as you're developing it and putting it together and trying to work on specifying what we've long called non-specific effects. So really some very interesting work. I'm going to turn this over to Bill Elwood to moderate our question and answer. For those of you who don't know, Bill coordinates these monthly webinar series. So that's always much appreciated, and to remind people that you can ask questions on the Q&A section as we move forward. So Bill, I'll turn it over to you.
Hey, Vitaly, could you speak, let's go to Neil first. Do you have any idea of how fMRI analyses, especially in the frontal lobes, might be hyperscannable?
Can you repeat that? What would be in the frontal lobes hyperscannable?
It's fNIR analysis.
Oh, fNIRS, functional near infrared spectroscopy?
Yeah.
Functional, yeah.
Yeah, so in addition to EEG, fNIRS has also been applied for hyperscanning, absolutely. In fact, we have an fNIRS group that's here at our center, and one thing I didn't mention is that we are working closely with them and hoping to do a spin-off project where we're actually going to be collecting EEG and fNIRS data simultaneously and looking at concordance with that data. So fNIRS is definitely has some advantages to fMRI. It also works with hemodynamic response. So it's not directly related to neuronal activity, but is indirectly related, similar to functional MRI. But it has the advantage of much improved temporal resolution, but also suffers from reduced spatial resolution. So there's kind of advantages and disadvantages of all these different neuroimaging technologies. But fNIRS is very interesting, and also in conjunction with the EEG response, I think we'll learn a lot by incorporating that into this type of hyperscanning research.
Excellent, thank you. You acknowledge your counterparts in Korea, but the open source software with facial recognition of emotion seem to be rather Euro-American centric. Is there concordance in facial expressions universally, or do you need to account for differences between your American and Korean research participants?
That's a good question. So first of all, just to clarify, the research is actually being done here on individuals. By here, I mean Boston. By individuals in Boston. So the Korean side of it is in expertise in some of the technology developments, data analysis, but we're not actually running any of the study in Korea. Having said that, there are absolutely Asian subjects, both on the acupuncture side and on the patient side, that are in the study. And the software that we're using was developed at MIT, actually. It was a spin-off out of the MIT Media Lab. And they actually incorporate that into their code, where there's some sort of automated tracking of culture and, culturally acceptable display rules that different cultures have, be it, Africans or Asians or Western Europeans, Americans. And so this is kind of taken into account, I think, by the software. But that's definitely a good question.
Oh, okay.
Yep.
Go
ahead.
Can I jump in?
Yeah.
So, Vitaly, this is Bill. I was thinking about the design that you have, and that the clinicians in these situations have some, at least, perceived sense of control over the patient's distress, right? But in most common patient-clinician interactions, they mostly listen and then prescribe. What are your thoughts about what would happen in the situation where they don't have either much control or feel like there's less perceived control over the ability to be able to affect the patient's levels of distress?
Right. So in some ways, that's akin to our control condition here, where we have a condition where the acupuncturist is actually instructed not to treat the patient. So they press a different button when they're not treating. And I didn't show this data, but actually there's reason to believe that the brain response by the clinicians when they're not treating, it can actually be stronger than the brain response when they are treating. And so there's kind of this, they have this empathy, and they want to treat the patient, and yet they're being told not to treat the patient. And so this sets up this whole interesting kind of like discordance. So, I mean, I guess luckily we have that aspect already built into the study because we have this non-treatment condition. And maybe what we need to do is actually look at concordance during the not-treatment condition in comparison to concordance during the treatment condition to see if there's any differences there and if that somehow impacts. The other thing is I mentioned before about the spin-off study that we're going to be hopefully starting in the next year with EEG and fMRIs. And in that case, we're actually going to be taking our data less so from the treatment condition, the treatment context, and more from an interaction context during the intake, which I think maybe is more, more generalizable to other clinician-patient interactions, because all clinicians and patients, go through an intake procedure. And so if we're taking data from that procedure and looking at concordance there, so that would be sort of similar to what the autonomic concordance studies that I introduced before by Marcy et al. Because that was more of like an intake or a interview that in that case the psychiatrist was undergoing with the patient. So I think this would be more akin to that, and that should yield interesting information too. So we're looking forward to that.
Good. Great. Yeah. Thanks. Okay. So let me just check if there are any other questions before we wrap up for today. I don't see anything else on the screen. Vitaly, that must mean that you were extremely clear and understandable. I
certainly think so.
Yeah, I think we all enjoyed it. Presentation is much appreciated. Again, we want to thank all of you for attending. Thank Vitaly again for his presentation today. And as I said, this presentation will be available online for us in about a month. So, Vitaly, thank you again for taking the time to do this.
Thank you. Absolutely. It was my pleasure.